This is LATHA AKULA. In this video, we are going to study about the plant-water relations. So, plant-water relations. We know that water is the essential element for all the living organisms. It may be a plant or animal or anything. Water is the important, a key role in all physiological activities. So, if we see in plants, in photosynthesis, water it plays a major role. Without water, there is no photosynthesis. Not only that, during photosynthesis, oxygen is released. That oxygen is released from the water only. If we see some examples, watermelon, it consists of nearly 92% of water. And in herbaceous plants also, they consist of only 10 to 15% of dry matter. Remaining percentage is 80 to 85%. It is occupied by the water only. And in mature corn plants, they absorb nearly 3 liters of water per day. And whereas the mustard plant, they absorb their weight per 5 hours. They absorb water equal to its own weight for about 5 hours. So that means for all the plants, the water plays important role, and this water acts as a limiting factor for the productivity and also for the growth of the plant in the agriculture. Next concept is water potential. Here, what is the water potential? We know free energy. In physics, we know what is a free energy. Free energy is nothing but potential to do work. That is called as free energy. Water is also have some free energy. That free energy of water is called as water potential or kinetic energy. And if you see, the pressure also acts as another source for the energy. And if the pressure increased for the water, the free energy of the water also increases, and that leads to increase in the potential. Water potential also increases when the pressure was increased. And how we can define the water potential? Water potential is also defined as it is nothing but chemical potential of the water. So chemical potential of the water, that means if in a system greater amount of the water is present, more amount of the water is present, then what will happen? Its kinetic energy or water potential also will be greater. That means if the water is greater, and its water potential also increases. And water potential, it is denoted by a symbol called psi. How it is denoted? It is denoted by psi, and it is expressed in pascals. It is expressed in Pascal units. And water potential of pure water, for example, if we take pure water at standard temperature without any pressure, the water potential of that pure water is 0. What is that? At the standard temperature without any pressure, the water potential of the pure water is 0. It is taken as 0. Next one is solute potential. What is the solute potential? Now we have seen water potential, which is denoted by the psi W. Now we see what is the solute potential. For a pure water, we have added some sugar, then it will become the solution. What is the solution? Solvent plus solute gives rise to solution, right? If some solute is dissolved in pure water, then it will become the solution. If you want to make a sugar solution, what we'll do? We'll take the pure water, to that some spoons of the sugar is added, then it will become the sugar solution or sucrose solution. And here, this is solution. In that solution, the number of free water molecules or concentration of the water decreases. If the number of free water molecules decreases, what will happen? The water potential also reduces. For a solution, the water potential reduces because of the addition of the solute. So because of the addition of the solute, the number of free water molecules or the concentration of the water decreases. That finally leads to the decrease in the water potential. When compared to the pure water, all solutions have low water potential. So all solutions have low water potential than the pure water because of the addition of the solute. So how we can define the solute potential? Solute potential can be defined as the magnitude of lowering due to the dissolution of the solute. That is called as solute potential. That means what? By adding solute, we have decreased the water potential. That is called as solute potential. That solute potential is denoted by the symbol psi s. How it is denoted? By psi s. And here this psi s is always negative. It is always negative. If more and more amount of the solute molecules are increased, then what will happen is finally the solute potential also becomes more negative. If more the solute molecules, the more negative the solute potential is. And at atmospheric pressure, at atmospheric pressure, if we take a solution, both the water potential and the solute potential, they both of them were equal. In which condition? At For a solution at atmospheric pressure, water potential and solute potential are equal. But instead of atmospheric pressure, we have added some more pressure. We have applied greater pressure than the atmospheric pressure then to a solution or pure water. Then the water potential also increases. In the water potential we have studied, right? So pressure gives the energy for the water, and finally it gives the increases the water potential. Here also the pressure greater than atmospheric pressure is applied to a water or a solution, then the water potential of that solution or pure water also increases. If we take a plant, to that water we pour some water, what will happen? The water will be absorbed by the roots and water will enter into all the cells of the plant, right? So the water molecules will diffuse into the cells. What it will causes? Because of the diffusion, that causes pressure build up against the cell wall. Because of the diffusion of the water molecules, some pressure will be built up against the cell wall, and it makes the cell becomes turgid. Turgid means swell. So the turgid cell, inside the cell, the protoplasm is present, right? That protoplasm also applies some pressure against the cell wall. The protoplasm also applies some pressure against the cell wall. That pressure is called as pressure potential. What is the pressure potential? The pressure applied by the protoplasm against the cell wall, that is called as pressure potential. This pressure potential is also called as Targar pressure, and it is denoted by psi p, and this psi p is always positive. We studied that solute potential is always negative, but this pressure potential is always positive. If we see the water potential is determined by the two components. They were solute potential and pressure potential. Solute potential and pressure potential are the two main components that determine the water potential. Then what is the equation? Water potential is equal to the solute potential plus pressure potential, that is psi w is equal to psi s plus psi p. This is very important equation. What is the water potential is equal to solute potential plus pressure potential. The next concept is osmosis. This is the cell. It consists of cell wall, cell membrane, right? Here this is cell wall. The plant is surrounded by the cell wall. It is freely permeable to water and other substances. It will not act as any barrier. But the cell membrane and the tonoplast. What is the tonoplast? In the cell wall there is a vacuoles are present, right? The vacuoles are surrounded by a membrane. That membrane is called as tonoplast. The cell membrane and tonoplast, these together play important role in the determining the movement of the molecules. So the cell wall will not act as a barrier, and who will determine the entry and exit of the molecules? That is cell membrane and tonoplast. Now we'll see what is osmosis. What is osmosis? Osmosis is nothing but the passage of the solvent molecules from the region of higher concentration to the region of lower concentration through a semi-permeable membrane. That is called as osmosis. And that osmosis is governed by the two factors. One is the concentration gradient and another one is the pressure gradient. For this osmosis we have one experiment. We will study about this. So this is the thistle funnel experiment. So here in this thistle funnel experiment, what we will do is we will take the funnel. That funnel is filled with some sucrose solution, and then to the mouth of the funnel, wrap it with a semi-permeable membrane, and then the solution is marked, and then this total funnel setup is placed in the beaker containing pure water, and then after some time the level of the sucrose solution in the funnel was observed. So after some times what will happen, the level of the sucrose solution raises. So what happened here, osmosis occurs. So in the osmosis what we have studied, the solvent molecules move from the region of higher concentration to the lower region of the lower concentration. So here the concentration of the solvent is higher, right, is more than the concentration of the solvent in the funnel. Hence the solvent molecules move from the region of higher concentration to the region of the lower concentration by crossing the semi-permeable membrane. Hence, the level of the sucrose solution also increased. That is why the level of the sucrose solution increased. But to prevent the entry of this water molecules, we have applied some pressure from the top. So, we have applied some pressure. So, more the solute molecules present in the funnel, more the pressure we have to apply against it. This pressure, what is osmotic pressure? The pressure applied to prevent the entry of the solvent molecules into the funnel, that is called as osmotic pressure. This osmotic pressure is always positive. It is always positive, and which is denoted by pi. And here there is one more thing is present. This is the osmotic pressure, and another one is the osmotic potential. This is also important. What is osmotic potential? Here the water molecules in the beaker, they also have the some potential, right? So the potential of these water molecules to move from hypotonic solution to the hypertonic solution through a semi-permeable membrane, that is called as osmotic potential. Here what is meant by hypotonic solution? The solution whose solvent concentration is high compared to the solute concentration, that solution is called as hypotonic solution. And what is a hypertonic solution? The solution where the number of solute molecules are more when compared to the number of the solvent molecules. Here the solvent concentration is more here, here the solute concentration is more here. This is the hypotonic solution, and this is the hypertonic solution. Here in the osmotic potential, what we studied, the capacity or the potential of the water molecules to move from the hypotonic solution to the hypertonic solution, that is called as osmotic potential. This osmotic potential is always negative. This osmotic potential is always negative. We have already studied, right? I have already explained about the experiment. This is the notes about that thistle funnel experiment and osmotic pressure and osmotic potential. So what I have explained about the osmotic pressure, that is the pressure required to prevent the entry of the water molecules from diffusing, that is called as osmotic pressure. It is always positive. If the more amount of the solute concentration is present in the funnel, the greater pressure is needed to prevent the water from the diffusing in. That is also we have studied. And what is osmotic potential? The osmotic potential is nothing but the potential of the water to molecules to move from the region of hypotonic solution to the hypertonic solution by crossing a semi-permeable membrane, that is called as osmotic potential, which is denoted by the psi s, and this osmotic potential is always negative. Numerically, osmotic pressure and osmotic potential both are equal, but the signs of these osmotic pressure and osmotic potential are opposite. So osmotic pressure is positive, osmotic potential is negative. This is the equation for this, that is psi s, and psi s is equal to minus pi.